Hey, good afternoon. My name is Haley Aber, and this is my PowerPoint presentation for Biology 1013, Mr. Siemens class. So let's go ahead and get to it. So I decided to name my title Banana, Blueberry, Pineapple, or Strawberry. Which one reacts with proteases activity? This is a picture of me. I just was super excited to do this experiment, and I love pineapple, so I had to put a photo in there. So we're going to go ahead and this is the outline. So we're going to do the introduction, background research, materials, methods, the experiment itself, the results I came up with, and a conclusion. Let's jump into the background. The introduction. So the question was, how can I determine if the fruits I choose to experiment on have proteases? Basically, whether they form into a solid or if they stay in a liquid form. Well, if they do have the proteases enzyme, they will be in the liquid. If they don't, they will be solidified. So my hypothesis was when I add fruits like blueberry, banana, pineapple, or strawberry to Jell-O, they should be able to solidify after being in the refrigerator overnight. This is a testable experiment, kind of just to see whether or not this was, I could test it, see if it's true. The purpose is to obviously evaluate and determine if any of the fruits I choose for this experiment, except for the pineapple, because we kind of already know that, have any proteases activity. Then some of the research I did, we had to find out what an enzyme was, which we know that it is it's a catalyst that speeds up chemical reactions. A protease is actually also it's a protein digesting enzyme, and a bromelain is a chemical that has the protease in it. So the pineapple has the bromelain, which also has the protease. So they digest the proteins, and gelatin is made up of proteins. So when you add the two together, one reacts to the other. Therefore, there's no form available for the Jell-O. The methods I used were the clear plastic cups. My choice of fruit was strawberry, banana, and blueberries. They're some of my favorites as well, along with the pineapple, the Jell-O mix, and the bowls and utensils we had to use too. Next, this is a picture, and besides the bananas being a little brown, I'm trying to stay organized, keep everything labeled, and everything kind of looking nice, preparing for my experiment. So the method that I used was go ahead and labeled all the cups according to what they were going to be in. So I began with cutting all the fruit in little pieces, and such as the banana, strawberry, pineapple, and blueberry. And also one for the control, which is just the Jell-O, and the other one for the heat-treated pineapple. I proceeded to follow the instructions to make the Jell-O, and also in another pot for the heat-treated pineapple, I boiled the water, set the pineapple in there for about 10 minutes, took it off the stove to cool down a little bit, and then once everything was done, put the fruits in the cups, put my Jell-O in each one of them, let it kind of cool for a little bit. After it was done, I put everything in the refrigerator, and I observed and analyzed and recorded the data, periodically, just checking on how everything was forming. And then I came up with my results, which we will get into, and my conclusion, and to share with you. So here's a couple photos of my materials. To the bottom left is another picture of my cutted fruit. The middle is me stirring to dissolve the jello. The bottom right is the before jello. So the top right is my labeled cups along with the fruits and my control. And this is a couple of them on the top left after I had put them in the refrigerator. This is all of the fruits, what they look like before the refrigeration. As you could tell, they're all in a liquid form. This is a few of them after. This is the jello, and all of them are tilted, so I know it might be a little bit hard to tell, but they're all tilted. As you can see, there's no liquid coming from the sides, which means that they're all in a solid form, meaning there is no protease in the fruits or in the control. The only one that did was the fresh pineapple, and basically because the bromelains were in it. I think the next slide, oh nope, this is the pineapple that was heat treated with the protease denatured by heat. Next we have my results. So the material used, I have all of my fruits and control on this area. The jello formation, you can see all but the fresh jello did form into a solid. And then, so the protease activity, there was none in the control. There was protease present in the fresh pineapple. Because of the heat, the protease was denatured in the heat treated pineapple, and in the rest of my fruits, there was no protease present. This is a video I'm gonna play real quick of, oh no, a video showing my all the fruits in their forms. In conclusion, so all of the fruits that I chose, none of them had the protease in them. Neither did the jello, or neither did the jello because it had gelatin in it, which was made up of protein. So the bromelain in the pineapple was the only one that was liquid, showing that there was absolutely some protease activity present. I had a lot of fun doing this presentation and this experiment, and I really hope all of you guys enjoyed watching, and I also hope that y'all have a great Thanksgiving. This is my boyfriend Dave. He also helped me do a little bit behind the scenes, so wanted to give a shout out to him. Well, that's it, y'all. Y'all have a great day. Thanks.